Okay, thank you very much. I will go right in the topic. The title of the presentation is When Evidence is Not What It Seems, Complexities in the Assessment of Skin Antiseptics. And this is a combined version of two talks. One I gave in a presentation in a conference in France, and the other one is in Geneva in Switzerland shortly thereafter. And so I have combined the slides a little bit to show improved version. So let's go into the second slide. I have no conflicts of interest to declare. And we will go to the third slide, History of Antisepsis. I will show two important people. One is Ignaz Semmelweis, who was a Hungarian obstetrician working in Vienna in Austria. He implemented hand antisepsis, and hand antisepsis effectively is a reduction of microorganisms on hands, which effectively laying the foundation of modern hand hygiene in healthcare. The second one is Joseph Lister, British surgeon who implemented wound antisepsis, which again became the precursor of skin antisepsis. Now, a bit of definition of skin antisepsis and relevance. Antisepsis is defined as the disinfection of living tissue, for example, hands and skin, healthcare workers and patients. The purpose of skin antisepsis is to reduce the number of microorganisms on skin, and the aim of this is to reduce the risk of infection or contamination from healthcare associated skin breaks such as incisions, punctures, etc. Now, I looked a bit into history, and here's an article from 1903 that shows that skin antisepsis is already prevalent in the early 1900s. This is an article from the Boston Medical and Surgical Journal in 1903, which is the precursor of the New England Journal of Medicine by authors Harrington and Walker. Now, this talk is associated with a bit of personal history and a personal experience, if I might say so, because starting around 2010, several colleagues and I noticed some unusual things in the literature. And so what happened was that several articles compared trial outcomes, clinical trial outcomes from chlorhexidine alcohol combinations, which is two active ingredients, to povidone iodine alone, which is one active ingredient. And these articles concluded chlorhexidine is better than povidone iodine. And several of us, several colleagues and I scratched our heads and thought, it doesn't make sense. We wrote several letters to the editor. In fact, it was a bit depressing because our letters to the editor had a little effect, and we thought something is wrong here in the literature, and we pursued this further. Now, to show you what is wrong in the literature or was wrong in the literature, I'm showing the slide, the antimicrobial spectrum and activity of skin antiseptics. This goes back to Elaine Larson's guideline on topical antimicrobial agents, 1988, and the CDC guideline 1999, the surgical guideline, and this is a composite figure taken from those two guidelines. So effectively, alcohols are said to have the most rapid activity and most profound antimicrobial activity. Chlorhexidine is less potent, but has excellent residual activity on skin. And iodine or iodophores also less potent than alcohol and have some residual activity that is considered minimal, meaning less than chlorhexidine. And if you combine chlorhexidine alcohol or iodine alcohol, these combinations have advantages such as additive effects and persistency. So in view of these problems in the literature, the questions arose, what is the factual evidence for chlorhexidine alone or chlorhexidine combinations in skin antisepsis? Then how common is the attribution of study outcomes to chlorhexidine alone when in fact a combination has been used? And the third question was, could this phenomenon that we observed have unjustly skewed evidence-based guidelines? Now, we employed a systematic review strategy. We did an exhaustive search for primary and secondary literature for clinical trials and systematic reviews, chlorhexidine versus competitors in skin antisepsis for blood cultures, intravascular catheter insertion and surgical skin preparation. And our criteria for literature assessment were twofold. One is attribution of study outcomes, meaning from alcohol plus chlorhexidine to chlorhexidine alone. And I will show you the principle in one of the next slides. And the second criterion was the factual evidence for chlorhexidine, which we determined in meta-analyses. And then we had a look at guidelines and narrative reviews to see if any of these misinterpretations or attributions problems spilled over into guidelines. Now, if you look at slide 9, you have a potential scheme of a clinical trial. So let's look at trial arm A. Trial arm A in this example gets two active ingredients, active ingredient 1 and active ingredient 2. And after the trial, there's clinical outcome A. And trial arm B has one active ingredient, in our case povidone iodine alone, and has clinical outcome B. So that's a common scenario as it was observed in trials at the time. So let's move to slide 10. Our criterion for assessment in our systematic review was the attribution of study outcomes from chlorhexidine plus alcohol to chlorhexidine alone. So if you look at this scheme here, this is articles concluding outcome A is caused by ingredient 1, or ingredient 1 is superior to ingredient 3, or the evidence supports ingredient 1. And as you can probably clearly see from this scheme, such conclusions are illogical and cannot be made. But nevertheless, those conclusions occurred relatively widely in the literature. Now, slide 11 shows the systematic review that was then subsequently published in PLOS ONE in 2012. And in slide 12, I will show you some of our main findings that we made. So we found good evidence that chlorhexidine plus alcohol is better than povidone iodine on its own, meaning povidone iodine aqueous, for all three applications: blood cultures, vascular catheter insertion, and surgical skin antisepsis. However, at this point in time, chlorhexidine plus alcohol versus povidone iodine plus alcohol, meaning two antiseptics versus two antiseptics, was either equal or inconclusive. I will get to that a little bit later. Subsequent to our review, it turned out that indeed for vascular catheter insertion, chlorhexidine plus alcohol is better than povidone iodine plus alcohol, but that is now, up to this time point, the only application where chlorhexidine plus alcohol is definitely superior to povidone iodine plus alcohol. Again, then point 3, we found no evidence for chlorhexidine alone for blood cultures and surgery for superficial skin. We found some evidence that chlorhexidine aqueous was better than povidone iodine aqueous for catheters, but the weight of evidence in total supports the use of chlorhexidine plus alcohol for catheters, which is better. But the striking finding in our systematic review was that an incorrect attribution of trial outcomes was common. So between the three different applications that we investigated, an average of 34% of attribution was incorrect, and a number of articles had ambiguous or unclear attribution, meaning an average of 54% of articles were not really clear on what had caused their trial outcomes. Furthermore, the antiseptic composition was often incomplete or missing. So only about 35% of all clinical trials and systematic reviews both correctly listed the antiseptics that had been tested, and correctly attributed efficacy to the components used. This is fairly substantial finding. And it did lead to unsubstantiated recommendations in clinical guidelines, which I'm not going to talk about in detail now because there are other things to talk here. If we go to slide 13, a few further downstream observations from the systematic reviews were that about 65% of clinical trial and systematic review authors were not fully clear or perhaps not fully aware, I don't know, of what it was that they actually tested. So again, I repeat that sentence. About 65% of clinical trial and systematic review authors in that area were not fully clear of what they had actually used in their trials or investigated in their systematic reviews. This is a fairly substantial finding. And substantial misinterpretations of evidence occurred despite the fact that most clinical trial and systematic review authors adhered to formal evidence analysis and reporting requirements. What it means is that the cookbook style adherence to formal evidence reporting requirements did not prevent these misinterpretations from being made. So my personal conclusion at this point is that knowledge of the subject matter at hand is a necessary prerequisite for assessing and interpreting evidence, which is a fact that is often overlooked. So let's go to slide 14. We made some further observations of errors, pitfalls, misinterpretations in the assessment of skin antiseptics, and we published these in one commentary style article in the Journal of Antimicrobial Chemotherapy and a letter to the editor in Infection Control and Hospital Epidemiology. So let's go to slide 15. One of these sources of errors in the assessment of antiseptics is the overlooked importance of neutralizers. And neutralizers have the function of stopping the activity of antiseptics after the regular exposure time to these antiseptics in standardized tests. So we have standardized microbiological antiseptic tests. There are US standards and European Union standards, and it is a requirement in both these standardized testing environments to add neutralizers so that after the exposure, continued killing of microorganisms is stopped. It is necessary for all antiseptics, but chlorhexidine is particularly affected because chlorhexidine exhibits strong bacteriostatic effects. And so a failure to include appropriate neutralizers can lead to false positive efficacy assessment, and unfortunately absent or insufficient neutralization is fairly common in the literature. So let's go to slide 16. We observed a number of other statistical and other inaccuracies and errors in the literature. There were frank miscalculation of statistics. There was inappropriate groupings of trial arms that were observed in the literature. So, for example, a trial when you grouped the trial arms for statistical analysis in a certain way, the authors showed significance supporting chlorhexidine, whereas if we grouped the trial arms in the classical way, there was no more significance. Or we observed selective reporting, unlike what you would expect to be reported. We observed inappropriate inclusion or exclusion of trials in meta-analyses, or we observed the assessment of trials with unknown or insufficient antiseptic content. And I will get to that later, because if you include trials where you don't know what you have actually used for antisepsis, you cannot make valid conclusions. And interestingly, the impact of the various errors and misinterpretations were disproportionately in favor of chlorhexidine. And let me say this, chlorhexidine is an effective antiseptic, there is no doubt about it, but the proportion to which it has been overestimated in the literature created a disconnect of its, how can I say, perceived efficacy with the real benefits. So the whole thing created a perception of chlorhexidine as the in-antiseptic. The reasons are often unclear. It may simply reflect a heightened interest of chlorhexidine researchers in their research topic, which happens very often. But there were also research scandals and alleged industry kickbacks. And Dr. Kavanagh knows about one of these instances where, in fact, a prominent patient safety advocate was involved in such a scandal. So let's go to slide 17. Now, on this slide, I'm making a bit of a provocative statement that is often overlooked. We need to be aware that the evidence for skin antiseptics does not arise solely from clinical trials out of a prior vacuum of information. This sounds very trivial, but this is part of the problems in the literature. So if you look at the US FDA drug development pathway, a typical pathway, you have basic research, you have compound discovery, you have preclinical development, like let's say lab testing, animal testing, etc. And then you have various phases of clinical trials. And so with all these information sources, you have multiple useful sources of information before or outside clinical trials that can inform you of important properties of antiseptics. And we should not overlook that. Let's go to slide 18. We have microbiological antiseptic testing. On the top of the slide, we have tests in reagent tube format. They are fairly simple tests, and shown is the qualitative suspension test. And on the bottom of the slides, we have tests under simulated practice conditions, such as tests on real skin or hands, etc., of volunteers. And we should stress that antiseptic testing has a history of nearly 100 years, and it has become increasingly standardized over the last 20 to 30 years. Let's look at slide 19. We have different antiseptic testing standards. So we have a series of US standards. The methods are described in the FDA Tentative Final Monograph 1994. The corresponding methods are published by the American Society of Testing Materials, and we have examples of a suspension test or a test on skin. We have also European standards. There used to be national member state protocols that are mostly unified in EN standards, and suspension test is an example, but tests on skin in Europe, we still have national tests only available. So let's look at slide 20. Here in this slide, I'm explaining the microbiological and clinical evidence for skin antiseptics. And we need to be aware of this difference. So microbiological testing clearly is a surrogate marker, and the clinical outcomes may differ. Yeah, we need to be aware of that. However, we know from about 100 years of history of testing shows that the results correlate reasonably well. In fact, I have not personally seen a single clinical trial result that contradicts known microbiological performance. So with microbiological testing, very detailed testing is possible, including optimization. So manufacturers commonly use microbiological testing to fine-tune the antimicrobial activity of their antiseptics. This is used for regulatory purposes, for example, US FDA tests. And because it's done on volunteers, there is no risk for patients from real infections. So on the other hand, clinical trials measure real clinical outcomes, such as catheter-related bloodstream infections, surgical site infections. This can be used for systematic reviews and meta-analyses and provides the strongest evidence to support clinical decisions. However, each test requires hundreds or perhaps thousands of real patients, which means it poses a logistical challenge. There is a risk from real infections, for example, catheter-related bloodstream infections or surgical site infections can be serious. So we need to be careful with clinical trials. And as a synopsis, as I said, I have personally not observed a single clinical trial result in history that contradicts the known or expected microbiological performance. So what it means is we have complementary sources of evidence here, and we should use these complementary sources of evidence wisely. Next slide, slide 21. In this slide, I'm explaining the principle of clinical equipoise. So clinical equipoise, also known as the principle of equipoise, provides an ethical framework for clinical trial research. So basically the principle is this: it should not be known before the clinical trial that one of the two trial arms is highly likely to be superior, because then we have a skewed situation whereby we're exposing one trial arm to an inferior treatment. Let's look at slide 22. So what are the ethical implications in antiseptic research? So we have several examples of trials in the literature, published trials of chlorhexidine plus alcohol versus povidone iodine alone. For example, I remember one trial published in the New England Journal of Medicine in 2010 about surgical site antisepsis. And if we look at the published microbiological literature, or if we ask any antiseptic experts, we could have made an informed guess of about a 10-to-1 performance difference in favor of chlorhexidine alcohol, mainly because of the alcohol component on the microbiological basis. Some of these trials involved serious clinical outcomes, such as catheter-related bloodstream infections or surgical site infections. And in these trials, more patients treated with inferior antiseptics had adverse outcomes. And to anyone in the antiseptic scene, that was no surprise at all. That's what we have expected. So the question is, was there clinical equipoise? I would say probably not. But interestingly, I would have normally have expected that this aspect is discussed somewhere, but it was not. So this aspect was not discussed in these trials, and no microbiological performance data were provided in any of these trials. Some of the trials were commercially sponsored. The question then comes up, is it likely that the companies were unaware of microbiological test results? And I would say no, because these companies would have tested their antiseptics before the trial as part of product registration. So it's not possible that the companies were unaware of microbiological test results. Then the other question comes, would a company have sponsored such a trial? Some of these trials were company sponsored. If the company had known that the antiseptics were microbiologically equivalent, and I would say maybe not. And so what we have here is we have another selective criterion that certain trials get done and published, and other trials maybe don't get done and published. Okay, let's go to slide 23. What is the microbiological performance? So if you look at the top left of the slide, chlorhexidine. Oh, sorry, I need to explain these are values from hand antisepsis. So we have pre-values, and then we have values at time point 0 after disinfection, and the further down it goes, the stronger the antiseptic is. And we have sustained activity after 180 minutes of under surgical gloves. So this is a classical slide from Professor Rotter in Vienna. And so if we look on the left, chlorhexidine is relatively weak on its own. Povidone iodine has less immediate activity than the alcohols, which are located toward the bottom. Alcohols have relatively best immediate activity. And if we look at chlorhexidine plus isopropanol versus isopropanol alone, the immediate activity is very similar. And then if we look at chlorhexidine plus isopropanol, then we see that chlorhexidine adds persistency to the mix. So, next slide, slide 24. What are the physiological and functional requirements? So, if we look at the three different applications, blood culture collection, surgical skin preparation, and vascular catheter insertion and maintenance, we see that the physiological requirements for persistency are quite different. Blood culture collection requires no persistency. Surgical skin preparation requires persistency for several hours, and vascular catheter insertion, due to its long duration, requires the biggest persistency. And that is basically a physiological explanation why chlorhexidine plus alcohol performs so well for catheters, simply because chlorhexidine among the antiseptics offers the biggest persistency. So next slide. What we also know is that different povidone iodine preparations exhibit different microbicidal performance, and it depends on how well stabilized and standardized the commercial formulations are. And it has to do with the amount of free versus complex iodine, and it means that the clinical performance for different preparations may differ. So it is not necessarily possible to extrapolate from a clinical trial and say, okay, all povidone iodine preparations perform the same. So let's look at slide 26. So we have here a slide using the FDA method to assess skin antiseptics. And what we see here is the baseline, then the reduction underneath the baseline of the microorganisms at 30 seconds, meaning immediately, at 6 hours and 24 hours. And if you look at the red dots, this is iodine plus alcohol, and the green dots, chlorhexidine plus alcohol. And interestingly, immediately after application, the microbial reduction of iodine alcohol is somewhat greater than chlorhexidine alcohol. But then, due to chlorhexidine's bigger persistency, after a certain amount of hours, this levels out. I should say clearly additional data are required, but these are interesting observations here. So let's go to the next slide, 27. Here we have the World Health Organization recent guidelines, and the WHO panel recommends chlorhexidine alcohol for surgical skin antisepsis, and this is based on the meta-analysis presented in the figure. So WHO recommends this as a strong recommendation with low to moderate quality of evidence. And if we look at the next slide, slide 28, and if we look at the factual quality of the included studies, then we see a few interesting features. So we have one trial that has only 23% isopropanol in the povidone iodine trial arm, and anyone who knows about the properties of alcohols knows that 23% of isopropanol is insufficient. Then other trials have unknown alcohol contents. Other trials are small trials. One trial is interesting, which is the Tulin 2016 trial. And the Tulin 2016 trial was included in the WHO analysis even after the WHO formal inclusion period. So this is interesting because we have several trials in it with unknown antiseptic content, which are, in my opinion, not informative in this context. And we have one trial included last minute after the official inclusion period. If we reanalyze these data with only trials with known and appropriate antiseptic ingredients, what we found is we found a large trial by Ngai et al. 2015 that was overlooked by WHO, despite the fact that it was published before the latest 2016 trial. If we include this trial, then we find that the results are not statistically significant. And I'm actually proud to say that this updated meta-analysis is going to appear as a letter to the editor in Lancet Infectious Diseases in about a month from now, so it has been accepted. And here we can see the updated 2017 CDC infection prevention guideline for surgery. And this guideline, I'm happy to say, still recommends alcohol-based antiseptics for surgical skin preparation with either chlorhexidine or iodine. So here in this case, the CDC guideline got it right. So let's advance to the second from last slide. In my opinion, we need a holistic approach for assessing skin antiseptics. We need the knowledge of basic antiseptic components. What is it that we're using? What is known about the ingredients? We need to know about the clinical and functional characteristics. What are the characteristics of the intended application? Do we need persistency? Is there use on mucous membranes? We need state-of-the-art microbiological testing according to recognized standards. In my personal opinion, no product should enter clinical trials without microbiological test results. Then we need clinical trials according to state-of-the-art trial standards. The microbiological test results, in my opinion, should be on-trial registry sites, and we need to be cognizant of ethical implications. And finally, we need to look at biological plausibility. Do the findings fit with what we know, and do the findings make sense? So going to the last slide, where do I see areas for action? We need to improve basic knowledge about antiseptics in the infection control community. We need to strengthen the role of standardized antiseptic testing and make it a prerequisite before entering clinical trials. We need to conduct clinical trials in line with trial standards and be cognizant of trial ethics. We need to rigorously identify and disclose conflicts of interest, and we will need to remain vigilant for any skewing or misinterpretation in evidence assessments and practice recommendations. And at that point, I would like to come to an end and thank you and invite any questions that you might have.